Our next speaker is Dr. Ben Marsland. Ben is now I have to read this, sorry, I have to put my glasses on again. Completed his PhD in immunology in New Zealand, Otago University, and the Marsland Institute of Medical Research in Wellington, and then spent 14 years in Switzerland, first at the ETH in Zurich, and then at Cloetta as a Cloetta Medical Research Fellow at the University Hospital in Lausanne. And during that period, he received the ETH Latsis Prize, the I can't even say that, Leonard's Prize, and ERS COPD Research Prize. So, since 2018, Ben has been the professor in the Department of Immunology and Pathology within the Central Clinical School here at Monash University. And he also maintains a visiting professorship with the University of Lausanne in Switzerland, and also as well as a Vesky Innovation Fellow and NHMRC Senior Research Fellow. So his focus of his lab is actually revolving around the microbiome in the gut, lung and skin, and how it influences asthma, respiratory viral infections and lung fibrosis. So he's going to talk to us about why bugs in the lung matter.
Great, thanks. Thanks, Bron, for the intro and the opportunity to join you here today. I suspect you're all sitting there feeling quietly confident that you know yourself, but I think it's important that you realise that you're more microbe than you are human. We're talking some big numbers here. So you have 100 trillion microbes in your body. And 100 trillion, I can't really get my head around very easy, but it's 100 with 12 zeros behind it. Or it's 10,000 times the population of the whole world. So it's a massive burden of microbes which are resident on each and every one of us, whether that be the skin, the lung, the gut, and most of our barrier surfaces, or surfaces which are exposed to the environment. But things get a little bit more complicated when we think about DNA. So most of us probably learn that we get half of our DNA from our mum and half of our DNA from our dad, which is true. But if we think about the DNA and all the genes which are actually part of who we are, our body, we need to think slightly bigger again. So you might be familiar with this spot, the MCG, or the G to the locals, as I'm told. And this stadium has 100,024 seats in it. So if we were to try to give every gene of our own in our body a seat at the MCG, we'd fill the MCC members' stand, so it's around 20,000 seats. So a fraction of the stadium would be filled with our individual genes. But if we think about the genes which are part of our microbiome, then one stadium, the MCG, is not enough. In fact, we need 20 MCGs in order to give every gene in our microbiome a seat. Around 2 million compared to 20,000 genes. So you can kind of imagine, with this microbial burden that we live with but don't really think about much, it's having a massive impact on our body. And we know most of our insight into the microbiome comes from research into the gut, and that's certainly where the majority of our microbes reside. We basically know most of our information about bacteria, but there are also fungi, in many cases helminths or worms and parasites, viruses, phages, all types of microbes which reside within this environment. But we really at the moment know mostly about bacteria. And so it's been a couple of hundred years that we've known about this in the gut. In the last 2 or 3 decades, particularly the last 2 decades, the field has exploded as we start to understand what each of these different microbes does, not just to each other, but what they do to our immune system, and consequently what they do to developmental susceptibility to different diseases. We're even starting to understand now that the microbes in our gut influence how we think. They can influence our brain function and activity, they can influence depression, and a number of other types of diseases. And we're only just now starting to understand that microbes also impact upon the lung. And part of the problem with that is, if you look at textbooks, and this is from the latest edition of one of the medical textbooks that students in Switzerland were using, and the dogma for ever really has been that the lung is sterile, that under normal healthy conditions there are no bacteria there. And this has been a dogma which was really tricky to break. And it's only been 9 years now that we know that in fact even in healthy steady state there are microbes in the lung, and we're starting to understand what those microbes do, but it's a very new field. We don't have all of the answers or many of the answers at all yet. So that's where the research that we and others around the world are doing, but particularly here, this is what happens specifically for us at the Alfred. You can't really see the details, it doesn't matter, but people like Carmel, who's sitting in the audience and will be sitting up in the panel later on, receive bronchoalveolar lavage fluid, which is when a bronchoscopy is performed and fluid is washed into the airways and then sucked back out and we can measure things in it. We receive this solution in the lab, which is in the building next door, and then we split it into all sorts of different types of conditions which are optimal for looking at bacteria or looking at viruses or looking at fungi or our own cells. And we need to treat them in all these different ways, put them into different types of tubes in order to have the optimal setting for us to look at fungal DNA, for example, compared to bacterial DNA or human DNA. So this is part of a biobank that we're trying to generate here in order to start addressing some of the fundamental questions that we want to answer, which is how can we make lungs healthier? So we've done this in certainly lung transplant, which is what I'll talk about shortly, but we've also done this in young babies. And this is something that we published last year, trying to understand when does the microbiome form and how does it form. And so what this graph here shows is the postnatal age in months, and this is the formation of the microbiome. And I think what you can tell from that is the microbiome has already formed essentially within the first 2 postnatal months. So it's really fast. And then this is how we look at the microbiome. We sequence the DNA of the microbes. We can use computers to then discover or identify what those sequences mean, what type of bacteria are there, and then we can put them into these community structures in a computer. And this is a healthy community structure that we see in healthy babies, where each of one of these circles represents a different bacteria. The bigger the circle represents how much of that bacteria there is, and the number of lines between these bacteria tell us how much they're talking to each other. So this is a healthy community of lots of bugs represented in relatively even quantities, except for one guy sitting out here, which is Morixella, and whilst he's part of a normal community, he's actually a bad boy and doesn't play well with his friends. So he sort of sits out here until there's trouble, and then he comes in and has a fight. This is a classic bad microbiome, and you can see with these babies, there are a couple of big circles there, and that's what we call dysbiosis, is a bloom of one type of bacteria which dominates, doesn't play well with anyone, and that's when you start to have disease. So we think about the lung as needing to have balance. And a classic tree, you see, we talk about the lung, or we talk about phylogenetic trees of microbes, the different ones that live together. But then when you have an imbalance, you have too much of one in particular, it's like an infection, or we're starting to understand that in a bit more detail, and that's what we call dysbiosis. So this is what we're doing to try to understand that in detail now. We take the microbiome and we take the samples from the lavages. We characterize the microbiome with sequencing its DNA and then putting it into computers. But we're also looking at what our own cells are expressing. So in this lavage fluid that we get, we have microbes and we have our own cells. We split them out and we can see this is what the human's doing, this is what the bacteria are doing. Now this data is not to try to understand, but to get an idea. Each one of these columns represents one lung transplant patient. Each one of the colours represents a different type of bacteria. And you can see that we've got a sort of a spectrum of different types of microbiomes for these different individual patients. At this end, these purple bars here, these are the bad bacteria, so the classic infections that you'd see, and this is really classical dysbiosis. This is what we understand as bad. At this end, though, it's quite interesting. There are far more of the good guys. There aren't hardly any of the bad guys. But in fact, what we found out, that this is not a healthy situation, and nor is this. In fact, what we need is to have a balance, predominantly of the good guys, but the bad guys are playing an important role in shaping the overall signals that go to our immune system and keeping us healthy. And you can see that to an extent with this picture here. Just look at the bottom. So these are fibroblasts, so this is like remodeling in the lung tissue. This is in vitro cells in the lab. If we look at the baseline, this is the fibroblasts here. If we add stimulant to those cells, which makes fibrosis develop in the Petri dish, we can see this orange coming in. This is the fibrosis. Now, if we just add the bacterial community, which is this one here, which has all the good guys in it, but too many of the good guys and not enough of the bad guys, we see that we have even more fibrosis, or fibrosis similar to what we would get if we gave a pro-fibrotic signal. So in fact, having too many good guys turns out not to be the right answer, not what we were expecting at all. Now, if you have the bad guys, it's definitely a problem. You get breakdown of the tissue. So don't get fibrosis, you actually just get destruction. So we're starting to get some sense from taking the samples from the patients, characterizing the microbes that are there, going back into the lab and seeing what do those microbes do. And what we've learned so far is that you need to have a balanced community which includes some bad microorganisms to give sufficient signals to your immune system to keep it healthy and your lung. So the final slide is where are we going now? And this is where, although I haven't been named a nerd, I don't think, I'll try not to snort with excitement because this is where we're going. It's super cool. So we have this bit, which is obviously very important, the lung transplant patients. We get the lavage fluid, which is what we've been talking about and how we've been analysing it. This is what it looks like, which is also quite cool and could go on your wall, as the bacterial communities that we see. When we look at bioinformatically at what they're doing, we can see representation of different microbes. We can see the same with the host gene expression. So we're starting to get this detail of what's going on with the microbes in the cells. We're actually expanding this now. We're looking at heaps of other high-dimensional data sets, things like lipids and metabolites and proteins, which are part of this. And what's really critical as part of this huge data set is also the clinical data, and that's why our interaction with the transplant division is so important, and a clinical database that is well curated so that we have all of the clinical information that we can link in with all of this big data. And then we start using really cool big computers which can compute all of that information and start to form by machine learning or going towards artificial intelligence types of approaches, what are the signals with all of that data that can cluster different patient groups together, and how does that then inform us into what their trajectory is going to be over time? And so part of our goal is to understand what can we know at this point here, which is going to lead to this patient developing CLAD, so a prognostic type of biomarker approach, but also what are the things that are going on here with the microbes and the immune cells, and can we understand something there that tells us about why people develop CLAD, and could we develop novel therapies against that? So next time you're at the MCG, just remember you're full of microbes. You're more microbe than you are human.